Mhm.
yenye inaanzia hadi penye inaishia.
Mhm.
Mm.
Now, how many types of digestion did we talk about? Like the one which is involved in. Sorry, continue.
Yeah, we talked about the physical and chemical.
And chemical digestion, where we say that the chemical involves enzymes,
Mm.
and the physical one involves chewing.
Yes.
We also went ahead and talked about digestion in the mouth. Where we say that in the
Mm.
mouth, the food is chewed, it is mixed with saliva. So unakumbuka components za saliva?
Saliva. Tulisema ni kuna enzyme amylase.
Mhm.
Yeah.
Yes, alafu tukasema kuko na mucus bado na kuna water. Good. So I think we can continue then. So after the food has been chewed in the mouth and mixed with saliva, some starch will be digested, and then the food will be rolled into boluses, which are going to be swallowed through the process known as peristalsis. So when food gets into the stomach, there's a hormone that is going to be produced known as the gastrin hormone, and the gastrin hormone is going to trigger production of gastric juice, and that gastric juice tulisema iko na HCl, and we say that the HCl is going to kill the disease causing microorganisms or pathogens or microorganisms in the food. And also the gastric juice tulisema iko na enzyme pepsin, but we also say that the pepsin is produced in inactive form, which is pepsinogen. And the function of pepsin, we said, is to digest proteins into peptides. And remember, we said that once the food from the mouth gets into the stomach, the action of salivary amylase stops. This is because the salivary amylase functions in almost alkaline pH, but in the stomach, there is an acidic pH produced by the HCl, which denatures the salivary amylase, hence stopping the digestion of starch in the stomach. So we also say that the stomach is made up of circular and longitudinal walls, which contract and relax to bring about a process known as churning. And churning is the mixing of the food in the stomach together with the enzymes and the gastric juice. And then, of course, we say the product that is formed is known as chyme. So churning is the process. The product formed is chyme. We also went ahead and talked about we say that the stomach is lined with goblet cells, which produce mucus that prevent the digestion of the walls of the stomach by enzyme pepsin, since the wall of the stomach is made of proteins, and pepsin digests proteins into peptides. So the digestive system protects itself by, number one, it produces some enzymes in inactive form, and also number two, it is lined with goblet cells which produce mucus that prevent the digestion of the walls. Remember tulisema, if in case there is any contact of the enzyme with the wall of the digestive system, it's going to cause what we call the ulcers or the wounds in the stomach. So today I want us to we also talked about digestion in the duodenum a bit, right?
Yes.
Yes, good. But we say that in the duodenum, there's a production of two substances. We say that there's bile juice coming from the bile, and also there's pancreatic juice. But we say that the bile juice is produced by the liver and produced and stored in the gallbladder, and then it is going to be released in the duodenum. But we say that in the duodenum, bile iko na kazi mbili. Number one, it neutralizes the acidic chyme, isn't it?
Yes.
And we also say that it contains salts which are going to break down fats droplets or oil droplets into smaller droplets. What do you call the process of breaking down complex fats or complex lipids into simpler droplets of lipids?
Hydrolysis.
We called it hydrolysis emulsification.
Emulsification.
Because hydrolysis near carbohydrates, okay?
Oh yeah.
Like for example, complex carbohydrates such as polysaccharides into simpler carbohydrates. So we call the process emulsification. So let us talk about the function of the pancreatic juice. So tulisema apart from bile, also in the duodenum, there is production of pancreatic juice by the pancreas, and the components of the pancreatic juice include pancreatic lipase, like I'm showing there. And of course, these enzymes digest the food substrates which they are named after. So pancreatic lipase means it is lipase produced by the pancreas. And then pancreatic lipase digests or breaks down fatty acids into glycerol. So lipids. You remember lipids? Lipids are broken down into fatty acids and glycerol because we said the building blocks of lipids are fatty acids and glycerol. Well, we said we have one glycerol. By the way, were you able to watch the video that I showed you before we left we ended the lesson?
Yes, I watched.
How was it?
Very helpful.
It was very helpful, isn't it?
Yeah.
Good. So we also say that in the duodenum, there's production of pancreatic amylase. This is very easy. Amylase from the word it's salivary amylase. So amylase breaks down starch into maltose. So inamaanisha ile starch yenye haikumalizika kudigestiwa kwa mdomo inamalizikia kudigestiwa kwa duodenum. And this makes a lot of sense kwa sababu we do not chew food for a long period of time. We just chew food for a short period of time. So we cannot be able to have broken all the starch. So therefore it only makes sense when the body has another part where starch will continue being digested into maltose. So therefore, the duodenum is the part where pancreatic amylase continues with the digestion of starch into maltose. Then we have sodium hydrogen carbonate. Now, sodium hydrogen carbonate is a base or a salt. And the function of sodium hydrogen carbonate is to emulsify the fats or oils into small droplets, and also it neutralizes the acidic chyme. So remember tulisema kwa tumbo kuko na HCl. HCl provides acidity. So the acidic chyme is going to be neutralized. Kwa sababu remember in the duodenum, the enzymes are not going to function in an acidic pH. The pH must be neutralized and made alkaline. That is where we must produce sodium hydrogen carbonate so that it can neutralize the acidity in the chyme so that we can have the enzymes in the duodenum functioning well because they thrive well in alkaline pH. Then the other part, the other component of the pancreatic juice need trypsin. Now I want you to look at trypsin. Trypsin, just like pepsin, it is not produced in its active form. Kwa sababu trypsin also digests proteins into peptides. So ni kumaanisha zile proteins hazikumalizika kudigestiwa kwa tumbo, zinamalizika kuwa digested in the duodenum. However, these two enzymes are different. They are different because pepsin in the stomach functions in acidic pH, while trypsin here in the duodenum, it functions in basic pH. So therefore, the basic pH which is produced by the sodium hydrogen carbonate is the one that is going out to activate trypsinogen to trypsin. So just like the way pepsin is not produced as pepsin but pepsinogen, even trypsin cannot be released as trypsin. It has to be released in an inactive form, which is trypsinogen. Otherwise, if it is released in its active form trypsin, it might break down the walls of the digestive system. So it is released as trypsinogen, and then it is activated later into trypsin to assist in breaking down of proteins into peptides. So now the enzyme that converts enzyme trypsinogen to trypsin is known as enterokinase. So trypsinogen is converted to trypsin in the duodenum by an enzyme known as enterokinase. So enterokinase converts trypsinogen to trypsin so that it can now break down proteins into peptides. Adaptations of the duodenum. How is the duodenum adapted to its function? Number one, it has Brunner's gland on its walls to secrete mucus for lubrication of food. Remember we said all the digestive system parts, they must be lubricated. This is because we do not want an instance where the digestive system is going to be digested just like the food, or maybe it is going to be hurt by the food that is moving in there. So for easier movement of food and for prevention against digestion by the enzymes, every part of the digestive system must have glands that produce mucus so that they can be protected against digestion. So duodenum, iko na cells known as the Brunner's glands which produce mucus, and that mucus plays a very important role in lubricating the food for easier movement and also prevention or protection against digestion by the digestive enzymes. The other one is that the duodenum has crypts of Libucan whose cells secrete digestive enzymes for digestion of food. So in short, you don't even have to say it has crypts of Libucan. Just say the duodenum produces or secretes digestive enzymes for digestion of food. Hivyo tu. The duodenum produces digestive enzymes for digestion of food. The third adaptation is that duodenum, it is concentrated to the pancreas and the liver to supply the juice and bile respectively for emulsification of fats and digestion of food. So ni kumaanisha that the duodenum is connected to the pancreas, meaning the pancreas and the liver empty their digestive juices into the duodenum. So therefore, the adaptation here is the duodenum is connected to the pancreas and the liver to be supplied with pancreatic juice and bile, which are used for digestion and also emulsification of fats and neutralization of chyme, respectively. So, as an adaptation, the duodenum is connected to the pancreas and the bile, which contribute to the digestion of materials in the duodenum. So we can have a practical activity to demonstrate emulsification of fats. Now this one we are going to do when you guys come for the session. Now by the way, I've prepared the practical sessions. You should be ready for the practical sessions. So I have like 4 sessions ready. So tukifunga shule, when you guys are ready, we'll organize and we'll do the practical sessions. I feel like we can include this session about emulsification, which is the breaking down of fat into smaller droplets. So I'm also going to include this practical so we can have 4 sessions which we can do in one day. Our biology sessions are going to take place in one day. We're not going to do them in 2 days. Just one day will be enough, because the good thing about biology is that biology sessions or practicals do not take a long period of time. Plus again, preparing for them is not that complex. You don't need a lot of requirements for the practical, so haitakuwa stress sana. So we can also include this one and see how emulsification takes place. But basically we can go through that practical activity together and see how it's supposed to play out. So the aim of the practical is to demonstrate emulsification of fats. And remember we said emulsification is like breaking down of fats, and fats are lipids. Remember, lipids are divided into 2, that is fats and oils. So you can have oils or you can have fats, but when they are broken down into small droplets, we say that process is emulsification. So how can we demonstrate emulsification? By number 1, we can have a sodium hydrogen carbonate solution We also need to have cooking oil. We also need to have water. We need to have test tubes, ruler, and measuring cylinder. Procedure will be, we will take 2 centimeter cubic of cooking oil and pour it into a test tube labeled A and B. Then we are going to add 2 centimeter cubic of sodium hydrogen carbonate solution into test tube A and rinse the measuring cylinder. So tutachukua 2 centimeter cubic of sodium hydrogen carbonate tuweke kwa test tube A, alafu we are going to rinse the measuring cylinder. We are going to add 2 centimeter cubic of water into test tube B. So tuko na test tube mbili. Moja iko na sodium hydrogen carbonate, ingine tutaweka maji. Then we are going to shake the contents in both test tubes, and then of course kwa test tube A kuko na maji sasa na kuko na sodium hydrogen carbonate. Alafu kwa test tube B kuko na cooking oil na kutakuwa na water. So in one test tube A kuko na only water, kuko na sodium hydrogen carbonate and oils. In another tuko na water and the oil. Then we shake the contents and then we write down the observations. So observations will be, there'll be formation of a white emulsion, and that white emulsion will be formed in test tube A, where we are going to have sodium hydrogen carbonate. So you'll be able to see sodium hydrogen carbonate has broken down. It has broken down the oil into very many droplets, and then again ile test tube B yenye iko na maji, utapata the oil and the water will not mix. Utapata there will be formation of 2 layers. The upper layer will be the layer of the oil, and on the lower side, the lower layer will be the layer of water, simply because water cannot break down the oil. Okay, so but for test tube A, where we had sodium hydrogen carbonate will have broken down the cooking oil, and instead of having now a liquid, we'll have an emulsion. Kitu inakaa ni kama solid hivyo, but very small droplets. So we can include that experiment. Also, instead of using sodium hydrogen carbonate, we can use lemon. Tunachukua cooking oil and we squeeze lemon into it. By the way, you can do this experiment by yourself at home also. Ebu try this. If you have lemon, tafuta a container, a small container, you add cooking oil, and then after you add cooking oil, kidogo tu kama kijiko moja, then get lemon, cut 1/2 of the lemon and squeeze the lemon juice into that container, and then you shake, or yes, you shake that container. Yes?
Repeat again. Add cooking oil.
You take cooking oil, you put it in a container, kidogo tu kama kijiko moja ama kijiko moja na nusu. I would prefer if you have a colorless container ama a clear container, and then you take a lemon and you cut 1/2 of the lemon, you squeeze the juice inside the container that has the cooking oil, and then you shake. You'll give me the observation that you're going to make. It will be the same observation you would make if you used sodium hydrogen carbonate, kwa sababu lemon has an alkaline substance which is sodium hydrogen carbonate in it, which will act on the cooking oil and break it into very small droplets of the cooking oil, and that will be an example of how emulsification takes place. That is the reason why ushachukua maziwa ukaweka lemon. By the way, kuna mtu asha interact na lemon na maziwa, what happens?
Inakatika.
It is the same thing, because lemon has basic substances in it which is going to break down the milk. So similarly, ukichukua lemon umwagie ndani ya nini ya cooking oil, you'll observe almost the same observation. But also we are going to do that experiment using sodium hydrogen carbonate, and we are going to make our conclusion. So that is in order, isn't it?
Yes.
Good. Let us talk about digestion in the ileum. Now, digestion ends in the ileum, ending. But we do not say the digestive system ends in the ileum. Digestive system begins or starts in the mouth and ends in the in us. But digestion process, ile process ya break down food substances, it ends in the ileum. And in the ileum, there is also a process of absorption. So the other name for ileum is the small intestine. And I'm sure from the video that you watched, you saw the small intestine and some of the functions that take place in the small intestine. So the small intestine is also known as ileum, and in the ileum there is production of digestive juices which are going to break down food or different types of food. So the ileum has two functions, or the small intestine has two functions. Number one, it completes the chemical digestion of food. And number two, it absorbs the digested food materials. So chakula kinamalizikia hapa kudigestiwa, and then it gets absorbed into the body. Now this is because the ileum has very many adaptations that enhance, like we are going to see. It has properties that enhance absorption process. Okay? Yes. So let us talk about digestion in the ileum. Digestion in the ileum. So once food leaves the duodenum, it gets into the small intestine. And in the small intestine, there are very many enzymes which complete the digestion. The word here is complete digestion. Now, before we even talk about the digestion in the ileum, there's something I want to bring into your attention. When food is in the mouth, it is digested, and the component of food that is digested in the mouth is starch. And the starch is broken down into maltose. In the duodenum also, the remaining starch will be digested into maltose. And if you remember very well, maltose is a component of carbohydrates which is simpler because food carbohydrates are stored in plants as starch. So the starch has to be broken down into a simpler substance which is maltose. However, the body cannot absorb maltose. So that means maltose has to further be broken down into a simpler component which the body can absorb. So back when we're talking about digestion, not digestion, components of food, the chemical compounds of the body of food, we talked about breaking down food which are complex into simpler substances. Like for example, we said the building blocks of carbohydrates ni monosaccharides. And examples of monosaccharides we said ni glucose. So that means food substances such as starch can be broken down into simplest properties which are monosaccharides. And basically that is what happens in the digestive system. Our digestive system picks complex food substances and breaks down into the simplest form of that food, and then the body absorbs that simplest form of the food. So the reason why we're breaking starch into maltose is so that we can break it down even further into glucose, which needs to be absorbed into the body. So the body cannot absorb maltose, it can only absorb the simplest form of a food substance. Then when we talked about proteins, we said proteins are complex, but they can be broken down into the simplest form of protein, which is amino acid, which means the body cannot absorb proteins. It has to break it down into amino acids. So the final product of proteins which can be absorbed in the body ni amino acids. That's why we break down proteins into peptides, and then peptides will have to be broken down into a simpler form, which is amino acids, so that our bodies can pick up the amino acids and use those amino acids to form whichever type of protein our body requires. When you talk about lipids, lipids exist as fats and oils, but our body cannot absorb fats and oils. They have to be broken into fatty acids and glycerol because our body only absorbs food substances in their simplest form. Have that been understood?
Yes.
So therefore, this means in the small intestine, there is finalization of digestion, such that the enzymes which are going to be produced in the small intestine, their function is to finalize digestion. Protein ilianza ikiwa protein, ikaenda kwa tumbo ikakuwa broken down by pepsin into peptides. Hii peptide bado si the simplest form of a protein. So it has to further be broken down into amino acids. So we need an enzyme that will break down peptides into amino acids. That is why in the small intestine, we are going to have an enzyme known as peptidase, so that it can break down the peptides into amino acids. We broke down lipids into very small particles through the process of emulsification. So tukienda kwa small intestine, we will have an enzyme known as lipase, named after the food substance it is breaking down. So lipase will now break down the lipids into fatty acids and glycerol, so that the body can absorb fatty acids and glycerol. We broke down starch into maltose in the stomach and in the duodenum, sindio? So we therefore need an enzyme that can break down maltose into glucose, and that enzyme is going to be named maltase. So all the enzymes which are produced in the small intestine are enzymes which are finalizing the digestion process. So like for example, the intestinal juice in the small intestine contains the following enzymes. Enzyme number 1, I'm reading from what I've displayed. Enzyme number 1, we have maltase. So maltase catalyzes breakdown of maltose into glucose. Sasa hii glucose inaweza ikaabsorbiwa kwa mwili. And this explains the reason why when an athlete has maybe run or maybe been engaged or involved in a athletic activity, they are not given ugali ama githeri. They are usually given glucose. Why? Because glucose will be absorbed directly. Hakutakuwa na digestion. You see, when you give an athlete ugali, they have to break down the ugali into glucose, and that process will take hours. But when you give them glucose, that glucose will be absorbed directly into the body, and it will be used for the process of respiration. Itatumika to provide the energy to that athlete. That explains the reason why they are given glucose, because it is already in its simplest form and it will be absorbed directly and used into the body directly. So that is why in the small intestine, the enzyme that finalizes digestion of starch is maltase, and it breaks down maltose into glucose. Sometimes we can have sucrose. Remember we said some of the food substances that we eat, they give us sucrose. Like for example, if it is sugarcane, it will give us sucrose. Most of the fruits will give us sucrose. So the sucrose will be broken down in the small intestine by an enzyme known as sucrase. Sucrase. And it is going to break down sucrose into glucose and fructose. And remember, glucose and fructose are monosaccharides, so they can easily be absorbed by the small intestine. Tunafuata?
Tuko hapa.
Yeah.
Good. So peptidase breaks down peptides into amino acids. Hizi peptides zitatoka wapi? Zingine zitatoka kwa stomach, kwenye pepsin ili break down proteins into peptides. Alafu pia kwa duodenum, where trypsin broke down the remaining proteins into peptides. So those peptides will be broken down to the simplest form, which is amino acids, and then the small intestine can easily absorb amino acids and transport them in the body. Then we have polypeptidase. Now polypeptidase, it catalyzes breakdowns of polypeptides to amino acids. Remember we said based on how many types of peptides are connected, we can have a polypeptide. A polypeptide is made up of very many peptides, okay? Like for example, we have more than two peptides combined together. they will be broken down by an enzyme known as polypeptidase. So polypeptides will be broken down into amino acids by enzyme polypeptidase. Then we have enzyme lipase. Tumesema kwa duodenum, the fats and oils will be broken down into small droplets, and this of course increases the surface area for digestion of the lipids. Now the specific enzyme that will break down the enzymes, not the enzymes, that will break down the fats and oils into fatty acids and glycerol is enzyme lipase, which comes from the word lipids. So we're just taking the food substrate and we add enzyme suffix ase, so that we can have enzyme lipase, and its function is to break down lipids into fatty acids and glycerol. Then we have lactase. Of course, lactase, you can get the answer from the name. It breaks down lactose into glucose and glucose and galactose. Of course, lactose comes from milk. So ukikunywa maziwa, hiyo carbohydrate inaitwa lactose. It will be broken down by lactase in the small intestine into glucose and galactose. Now remember, glucose and galactose are small, they are the smallest forms of carbohydrates, so they are monosaccharides. So kindly note, digestion of food is completed in the ileum, in the small intestine. Now, that food which has been broken down into the simplest form and is ready for absorption is referred to as chyle. CHYLE. Chyme iko kwa stomach. Sasa even the final food substance that is ready for absorption is known as chyle, and that takes place in the small intestine. Now, adaptations of the small intestine which enhance it to, which make it makes it easy for the small intestine to absorb food. Now, remember we have said small intestine or the ileum has two functions. It has a digestive function and an absorption function. So the question is, how is it adapted to digestion and absorption? Tumesema kwa digestion iko na hizo enzymes zote zenye tumeongelelea, lipase, we have maltase, sucrase, peptidase. All those are enzymes produced in the small intestine for digestion. Now, how is the small intestine adapted? How is it made? An adaptation is a modification. Like, for example, we can say, let me ask a question here so that you can understand the meaning of adaptation. Let me first of all define adaptation. Adaptation is a modification of an organism to survive in a particular habitat or to be able to perform a specific function. Like, for example, let's talk about human beings. How are females adapted to their role? How are females adapted to their roles? Like, what is the role of a female in the population, in the human population mostly?
Reproduction.
Reproduction, to give birth, isn't it? And males don't give birth, but females give birth. So how are females adapted to the process of giving birth and raising the young one?
They have uterus which holds the baby, which carries the baby. They also have mammary glands.
Good. So if you look at the mammary glands of the females and the males, they are not the same. Females have advanced and more modified mammary glands because they are specifically modified for raising and suckling the young one, isn't it? When it comes to males, they have no uterus, but females have a uterus. So this is where the fetus is going to grow, the baby is going to grow inside the mother. So therefore you see, females are adapted to their purpose and males are not going to have that adaptation because they don't perform that function. So the small intestine therefore also has some features which are absent in all the other parts of the digestive system, primarily for the function of absorption. So what are the adaptations? So an adaptation is a modification so that a part of an organism or the whole organism can be able to perform a specific function. So how are how is the small intestine adapted to the process of absorption? Number one, it is long to increase the surface area for absorption of food. Ukianza chukua small intestine utaona ni refu. By the way, you guys eat matumbo?
Yes.
Yes, we do.
I
Matumbo
don't take.
inakuanga small intestine. Yes. The other day you said you took matumbo, eh? Now,
No,
matumbo
not me.
yeah.
Me don't take. Mm.
Oh, you don't take, but you see, you know the matumbo, isn't it?
Yes, yes.
Yes, hiyo matumbo ndio inaitwa small intestine, eh? So matumbo ukiona inakuanga refu sana sana, it is very long. The reason why it is long is so that it can increase the absorption of food, the absorption rate. How does it increase? You see, because food has to move along the small intestine, because it is long, it is going to take a long period of time before hiyo chakula itoke kwa small intestine. So unapata inafanywa ikuwa refu so that food will take a long period of time in the small intestine. And the more the food takes in the small intestine, the higher the rate of absorption. So chakula kitakaa sana hapo for a long time. So imetengenezwa ikiwa refu so that the food will take a lot of time in the small intestine, and in the process of taking long time in the small intestine itakuwa inakuwa absorbed. So the longer it takes, the higher the rates of absorption. Yes?
Can you repeat that point again?
Nimesema the small intestine is long. Ni refu. Now the reason why it is long is so that it can increase the time taken by the food in the small intestine. And the more the time, the higher the absorption rate, because the food will be in contact with the small intestine for a long period of time. So chakula kitakaa hapo for a long time, and the more it takes to be in the small intestine, the higher the absorption rate. So by the time inatoka kutoka pale mwanzo hadi mwisho, most of the food substance will be absorbed because it is going to take a longer period of time in the small intestine. Understood?
Yeah.
The other adaptation is that the small intestine is highly coiled to slow down the movement of food. So apart from being long, it is highly coiled. The reason why it is highly coiled is so that food itaenda pole pole. Now as food moves slowly, then the food will be absorbed into the body. So in short, being long and highly coiled, they are increasing the time that the food is going to spend in the small intestine. And this is going to be very important for absorption because the more the food will be in the small intestine, the more the chances that it is going to be absorbed into the body, because the food will be in contact with the small intestine for a long period of time. So both being coiled and being long, they increase the time in which the food will be in the small intestine, which increases the absorption rate. Sawa? The other adaptation is that the inner walls of the small intestine has hairs known as villi or microvilli. So I'm sure nyinyi wote mshaiona matumbo, si ndio? Na saidi ya ndani ya matumbo inakuanga inakaa ni kama iko na tunywele nywele. Yes, hizo ndizo tunaita villi na microvilli. So hizo zinakuanga projections zenye zinafanyanga absorption to take place. So unaona huto tu villi venye ni mingi hivyo, hizo tunywele tunakaa ndani ya small intestine, kazi yake ni kuabsorb food. So food inapitianga kwa hizo structures, so they are numerous so that they can absorb the food materials. So that is an adaptation. So ukiona matumbo next time, utu tunywele twingi tunakaa zaidi ya ndani, we refer to them as villi or microvilli, and their function is to increase the surface area for absorption of food. So that is why the small intestine is lined with numerous villi and microvilli, so that it can enhance or increase the rate of absorption of food substances. Sawa?
Okay.
Good. The other adaptation is that the small intestine has goblet cells. iko na cells za goblet zenye zina produce mucus, just like the stomach and the esophagus. The small intestine must be lubricated with mucus. That mucus has two functions. Number one, it allows food to move freely, and number two, it protects the walls of the digestive system of the small intestine from being broken down by the enzymes which are breaking down the food. So every part of the digestive system where digestion takes place lazima kukuwe na mucus. Hii mucus kazi yake ni prevent digestion of the wall of that part of the digestive system, and also to make the movement of food very easy by lubrication. Ndio maana wewe ukishakula chakula hutaisikia chakula ikimove kwa mwili kwa sababu it is highly lubricated. So in the small intestine also we have goblet cells which secrete mucus, which is going to lubricate the food and protect the walls against digestion by enzymes. And then we have the walls have glands that secrete intestinal juice which contains enzymes for the digestion of food. Remember tumesema in the small intestine kuna kazi mbili. Kazi ya kwanza ni digestion, kazi ya pili ni absorption. So zile enzymes zote zenye tumetaja ziko kwa small intestine zinaproduciwangwa ndani ya small intestine. So we say that the walls of the small intestine have glands known as the crypts of Lubberkan. So Lubberkan glands, they secrete intestinal juice. Na nimesema hii jina Lubberkan ikikusumbua, si lazima useme, unaweza tu sema that the walls of the small intestine secretes intestinal juices which break down the food substances to the final products ama to the simplest form. So si lazima usumbukane na hiyo jina crypts of Lubberkan, but ukiweza kumbuka it is still okay. But the most important adaptation here is the secretion of intestinal juice which finalize the digestion of food materials in the small intestine. The other adaptation of the small intestine is that it is narrow. It is narrow. Narrow means haiko widely spaced. So ile path yenye chakula inapitia ndani ya small intestine ni ndogo sana. It is narrow so that chakula ikiwa ndani ya small intestine iko in contact with the walls of the small intestine and the villi na microvilli. This allows absorption to take place very well. So kwa sababu unapata chakula yote yenye iko kwa small intestine iko in contact with the walls of the small intestine. Hii itafanya absorption ikuwe easier kwa sababu chakula already kiko in contact na wall, so food materials will move through diffusion from the small intestine into the bloodstream that is next to the small intestine. So the adaptation is the walls of the small intestine are narrow to increase the absorption rate of food. The other adaptation of the small intestine to its function is that the small intestine has highly vascularized walls. It has highly vascularized walls. Or we can also say the walls of the small intestine are well supplied with blood capillaries. Now, hizi blood capillaries ziko na kazi gani? Ukiangalia small intestine inakuwa surrounded by blood vessels kwa sababu wakati chakula kimekuwa absorbed kutoka kwa small intestine huwa kinapelekwa kwa blood system or the circulatory system, kwa sababu it is the blood that is going to transport the absorbed food materials to the parts of the body where the food is required. So kama your brain requires glucose for respiration, that glucose will be taken from the small intestine through the circulatory system. So therefore lazima small intestine ikuwe na blood vessels mingi, ndio chakula kikikuwa absorbed, that food is going to be transported to all parts of the body through the circulatory system. So otherwise kama hakungekuwa na blood vessels, chakula hata kikiabsorbiwa hakiwezi kikafika to different parts of the body. Therefore, the small intestine must have a highly vascularized wall, or it must have a very high network of blood capillaries which is going to transport the absorbed food from the small intestine to all the parts of the food. Very good. So The other adaptation is that the small intestine has smooth muscles which contract and relax to facilitate peristalsis. Remember tulisema peristalsis is movement of in the small ama in the digestive system. So ndio chakula kiweze kumove kwa small intestine. The small intestine is made up of muscles which are smooth muscles. Now remember tulisema smooth muscles line up the digestive system and they easily contract and relax to bring about the movement of food in the digestive system. So now small intestine iko na smooth muscles which are going to allow the food to move easily in the digestive system. Another adaptation number 1 is that the digestive system has lacteal vessels for absorption of fatty acids and glycerol. So unaona ile let me see if I can show you the diagram. Now, good. You remember the hairs which we've talked about? The ones which we've said which are found in the small intestine. This is how the hairs look like. This is the structure of that hair. That hair has a dense network of blood vessels. Ukiangalia side ya ndani utaona hizi parts zenye zinakaa red. Hiyo ni blood vessel. Na kazi ya hizo blood vessels ni chakula kikiingia ndani ya small intestine kitaabsorbiwa na the lacteal, not the lacteals, by the hair. So hii ni hair yenye inakuanga ndani ya small intestine. So chakula kikiingia ndani ya hiyo hair inaenda direct kwa bloodstream, alafu inakuwa transported to all parts of the body where it is required. Katikati ya hizo blood vessel kuko na structure moja hapo inaitwa lacteal. Can you see the lacteal?
Yes.
Now, every hair in the small intestine has a lacteal. Lacteal kazi yake ni kuabsorb fatty acids and glycerol. So lipids zinaabsorbiwangwa na hii structure inaitwa nini? Inaitwa lacteal. So zile fatty acids zilidigestiwa na lipase zinakuwa absorbed by lacteal. So therefore, another adaptation of the small intestine is that it contains lacteals which absorb fatty acids and glycerol. Sawa sawa? And then of course, in the small intestine, you're going to find numerous mitochondria, which will provide the energy for the absorption of food. Now, this section, this part here about the adaptations of the small intestine to its function, it is highly examinable. And this section is tested as an SC question. You remember the SC questions we talked about?
Yes.
Yes, so this is how they phrase the question. So I also want you to do this SC question. So kindly, when you get time, you do this SC question, you take a photo and you send it to me, and then I'm going to see how you have done this SC question. Sindio? So write down this SC question. The SC question is discuss the adaptation of the small intestine to its function. Discuss the adaptations of the small intestine. Discuss the adaptations of the small intestine. Discuss the adaptations of the small intestine to its function. Discuss the adaptations of the small intestines to its function. So I'll give you 10 marks for that. So in short, you're supposed to state each and every adaptation of the small intestine and state its role or its adaptations. Indio?
Yes.
Now let me guide you on how you're supposed to do an SC question. Number 1, every SC question must have a heading. So what will be the heading for that question?
Adaptation of small intestine to its function.
Yes, very good. Adaptation of the small intestine to its function. Now, when you're doing your essay, do not number your points. Okay? So how are you supposed to do it? You'd rather write those adaptations zikiwa kiparagraph kimoja. Unaanza unasema, the small intestine is highly vascularized has high dense network of blood vessels to transport the absorbed food, full stop. Immediately after that full stop, endelea useme it also has, unaona? So you'd rather write the adaptations ikiwa in one very big paragraph, or you can have your points as a paragraph, but usiandike number 1, number 2, number 3, ama A, B, C, D. ama uweke zile dots za kuonyesha ni points. It is highly discouraged. So you're going to have the heading and then you write the points. That is how you're supposed to do that question. So I think we're going to end the lesson there because we have now talked about that process. So when we meet during the next lesson, we are going to, that is today in the evening, isn't it? Ukuje ukiwa ready. Kila mtu nitampea chance yake atudescribie kenye understand ya digestion from the stomach to the small intestine, si ndio?
Yes, teacher.
This question, yes.
From the stomach, teacher.
Sorry, let's have answer. Domitila?
I was asking.
Is it from the stomach to small intestine?
No, from the mouth to the small intestine, sorry.
Okay.
Monica?
Teacher, I was asking. Tumeongea kuhusu the paragraph. Umesema we can write it, just one paragraph. Yote iko in one paragraph.
Yes, ikiwa kimoja kiparagraph kimoja kikubwa. That is even better.
Yeah. Then alafu when you are starting kama kwa English you have kuna hizo rules za kuandika paragraph. So this one should you start at the margin ama at the center ama any?
That you don't have to start at, what you do is answer two at the margin and then upeane. What you do is ukiweza ona unataka kuandika in several paragraphs, tuseme kama for example umeandika adaptations kama 4 na unataka kuanza paragraph ingine, just skip one line. Skip one line na uendelee kustate in paragraph form. Sawa?
Okay. Kama unataka kuziseparate hizo
kama unataka
points
kuziseparate but there is no need, you can just write them as one paragraph, hiyo kiparagraph kimoja kikubwa.
Okay. Sawa.
Sawa. So when we meet tonight, we are going to have a session where you're going to, don't worry, wewe utatuexplainia kenye umeelewa ndio nijue ni wapi tunafaa kurudia ama ni wapi tunafaa kuemphasize, si ndio?
Yes.
So utadiscuss digestion kenye umeelewa from the mouth all the way to the, and leo ni holiday, so I'm sure hardly will we go to work, and even if we go maybe tutapata chance kidogo ya kupitia, si ndio? So unaweza pitia hiyo digestion process kenye tu utaelewa, usikuwe na pressure, kenye tu utaelewa, alafu tukipatana you'll discuss for us, we will correct you where you have gone wrong, we will commend you for where you've gotten it right, and then we can comfortably do an essay question on that, because hii process ya digestion inakujanga ikiwa kama essay ama kama maswali. So after you have discussed that, nitatafuta maswali kadhaa yenye ina involve digestion na tuifanye. So leo itakuwa nyinyi muni explainie kenye mmeelewa kwa hiyo digestion, si ndio?
Sawa. Then the assignment unazihitaji leo?
If possible, yes, if possible you can send it today, ndio by jioni nitakuwa nimeweza kujua penye umekosea, tutaipitia juu kesho niko sure we'll do something different.
Okay.
So have yourselves a beautiful day. Take care of yourselves, sir.
You too. Thank you.
Okay, good day.
Okay, bye.